Hello and welcome everyone to today's webinar, The Adventures of RNA, a fun journey through purification. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Bioecho Life Sciences. To learn more, please visit bioecho.com. Now we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Additionally, we'd like to further interaction during the webinar, and therefore we will have several polls throughout the presentation. The results will be seen and discussed by our speakers. I'd now like to welcome our moderator, Christiane Schlattbaum, Director of Marketing at Bioecho Life Sciences, and she will be your moderator for today's event. Christiane, over to you.
Thank you so much, Christy, and welcome everyone to our webinar, also from my side. I'm Christiane Schlattbaum, as said. I'm a biotechnologist, joined Bioecho 3 years ago as Director of Marketing, and together with me is Thomas Winkelmüller. Hi, Tom.
Hi, nice to see you, all of you. My name is Thomas. I'm also working at Bioecho as a sales manager, and I'm really looking forward to the next 45 minutes to an hour to have a very interactive session with you. Maybe short introduction. So my background is in plant genetics, where I did my PhD, and for the last years, more than 6 years, I worked quite intensively in nucleic acid purification and technical support, now in sales. And I'm really looking forward to the webinar today.
So today we prepared a story for you, the story of an adventure of RNA from the cell to the reaction tube. Our webinar is going to be interactive and fun, hopefully, but also scientific and full of practical tips and tricks for you. Tom and I will be playing the ball back and forth a bit, me telling the story and Tom focusing on the scientific part. So we really hope you like our approach. And as Christy indicated already, we also prepared some poll questions for you in between to get your input. So I really do hope it's going to be an interactive session. In case you don't know us yet, Bioecho Life Sciences was founded in 2016 by leading industry experts, and we are a specialized solution provider for the extraction and analysis of nucleic acids. We strive to make extraction and downstream processing really easier and faster, significantly increase throughput, and deliver reliable results. And what is more, sustainability is part of our DNA. Summing up, we believe that even the most routine application in molecular biology can be improved. With years of experience, we bring innovation into molecular biology, and we handle all our projects with dedication, expertise, and flexibility to really be the nucleic acid experts for you. Our little story today begins in a hidden world for us, so tiny that we can only see it under the microscope. A typical animal cell is 100,000 times smaller than we are. To bring this in scale, the size of a cell is as small to us as we are to the greater Tokyo area. The journey of our RNA molecule begins in the heart of the cell, the nucleus. It all starts with the transcription. DNA, the blueprint of life, unwinds to reveal a gene that needs to be expressed. RNA polymerase, a crucial enzyme, binds to the DNA and starts to transcribe the genetic code into the RNA sequence. The hero of our story is born, the RNA molecule that during the next 30 minutes we are going to follow on its way from the cell to our reaction tube. Once transcribed, the RNA molecule is not quite ready to go on its journey and fulfill its job. Depending on the job it's supposed to do in the future, it needs to undergo several maturation steps, such as getting a protective shelf and items that help to make it rigid for the world outside of the nucleolus. For certain molecules, unnecessary parts are cut away so that there are multiple layers of editing already early on. During maturation, our RNA molecule recognizes that its friends are modulated in a very different ways and look quite different from him. One of its friends will become a transfer RNA, acting as a transporter and bringing amino acids to the ribosome for protein synthesis. This one will be responsible to read the code on other RNA molecules and match them with the right amino acids to build proteins. Another mate becomes a ribosomal RNA, which acts as the construction worker. It will be part of the cellular machinery that builds up proteins and will help to catalyze the formation of peptide bonds between amino acids that assemble into the protein. But how to know how to assemble all the important protein building blocks for our body? Here comes our mRNA molecule, who functions as the architect, instructing its friends how and what to build. Together, they form a team that is crucial to fulfill the right function of a cell. However, there are many more different RNA molecules.
Okay, so here it's basically the first time that we zoom out of our story, because before starting your RNA isolation, we believe that it's very crucial that you think about and that you know what you're looking for. So as we just seen in that nice example where we had the transport RNA and this architect messenger RNA, all of you are aware that we have all of these different RNA species that you see on the slide here. And as you could just see by easy look here, they all look quite different in structure, and they are also quite different in size. So for example, if we want to look at the microRNA, we have a very, very small molecule. And this, of course, has also quite important implications for the method that we want to choose for the RNA isolation. Another thing which is probably known to all of us, but it's easily forgotten is that most of the RNA that we will isolate will be the ribosomal and transporter RNA, which make up roughly 95% of the RNA, while only 3 to 7% of the RNA will be messenger RNA, which is a type of this molecule which many of us look for if we look for differential gene expression. So this is really important in the start before you choose your method to exactly know what you're looking for. So after the maturation of all these molecules, these are exported to the cytosol and actually start to fulfill their job. And this is where it becomes interesting for us as a scientists.
After the RNA molecules are released from the nucleus, they are happily doing their job, not suspecting anything bad. What they cannot anticipate is that some curious scientists wonder what's going on in their secret world. Suddenly, their world explodes, and our friends get pulled out of the cell. While the cell membranes burst, our RNA friends can rescue themselves and stay alive. But how are the scientists letting the cell explode without harming the RNA?
Okay, so now we really come to the first step of our RNA isolation, which is basically, first of all, we need to release the RNA from our sample and from our cell. And basically, we can divide an RNA extraction two major parts, with the first quest being the sample extraction, so releasing the RNA from the cells, and the second part that we will look a bit closer later on, being the purification of the RNA molecule. And as Christiane nicely explained in our story, although we make all these cells burst, surprisingly, the RNA stays intact, and this is quite important for us as a scientist. So we will now discuss a few methods for this, but before we do so, we have our first poll question. Please wake up, as we are quite curious to know, so what type of samples are you usually looking for when you're extracting RNA? So are you doing RNA extraction for blood, from culture cells, from tissue, FFPE plant, or even other sample types?
From your experience, Tom, with talking to many customers, what would you expect?
I guess that likely the majority of people are using cultured cell or tissue samples. At least this is kind of based on market research, the biggest numbers of isolations that are done. But I'm also pretty curious if some of the people in the audience maybe use some more exotic or challenging samples. Yeah, so maybe we can take a look at the results. Okay, and we can push that to the audience. Okay. Seems not that, at least to me, it looks like nobody participated. Now I see. Okay. Seems so either I have a lag in my internet connection because it looked like 0 replies. I was a bit shocked. But
No, we
now
do
it's
have
much
some
more.
results.
Yeah.
Yeah. So
Yeah, absolutely.
it's indeed similar to what we anticipated. So blood and cultured cells indeed being predominant,
Yeah.
but there's also large variety, as we can see. Nice. It's
Yeah.
cool to see
So
that we
some more
have
challenging.
such a mixed audience
Yeah.
today. Okay.
So today we won't focus that much on something like plant or FFPE, but if any questions to that come up, please feel always free to write them in the chat or ask them later on in a QA session. So this is also quite an important part of the webinar, of course. Okay, so let's go on. So as we nicely saw by the poll, yeah, you're looking at very, very different sample types. And this is the first thing where it's really important that we make sure that we choose a homogenization and lysis method which is specific to the type of sample that we're looking for. This is also one of the reasons why you will find many specific products and kits for certain sample types like a plant RNA kit or tissue RNA kit. And I would like to highlight some of the most important methods to perform the lysis and also explain you some of the advantages and disadvantages that these methods have. So you can evaluate a bit better which may be the perfect method for you. So first of all, we have chemical lysis, which is oftentimes using detergents or organic solvents. Also chaotropic salts are often used. Then we have enzymatic lysis using either quite, let's say, general enzymes like proteinase K or protease, but also sometimes more specific enzymes like lysozyme or zymolase. And of course, we have a large bunch of mechanical homogenization and lysis methods like free Thor cycles and using bead tubes or bead mills to really lyse the sample of your choice. So let's look at some of the advantages and disadvantages of these methods. So first of all, we have the chemical lysis. A typical example for that would be trisol, which is a quite common method to isolate RNA. So the advantages here, it's clearly that usually it's fairly fast, so we usually don't have long incubation times. And it's a pretty universal lysis oftentimes. So I can use it for a broad variety of different sample types, including also some more challenging sample types like plants or soil or something like that. But it comes also with some major disadvantages. And that's first of all, many times the chemicals are used either quite toxic, like phenol, or they are used in such high concentration that it's also toxic. So it's not that nice to work with that many times. We need to go under the hood. And obviously, these are quite strong PCR inhibitors. So when we introduce them to our isolation, we later on need to make sure to remove them again before we can actually use the RNA. The second example would be the enzymatic lysis. Here, the major advantages is it's pretty flexible because I can play a lot with the amount of enzyme, the concentration that I use. Usually, for enzymatic lysis, I need incubation at a certain temperature. I can also play with that incubation time to prolong that. if I see that I need a bit longer lysis times. And also, I can also achieve a quite specific lysis. So in the example of, for example, taking a lysozyme, this is an antimicrobial enzyme that is produced by animals as part of their innate immune system, which will basically is a glycoside hydrolyse, which will hydrolyze peptidoglucan, which is a major building block of gram-positive bacterial cell walls. So this will quite specifically lyse gram-positive bacterial cells. Some disadvantages, we need that time to incubate, so it's prolonging our procedure. It can be sometimes expensive depending on the enzyme. So usually enzymes are fairly expensive compared to, let's say, a salt buffer. And also we need, of course, some device to do the temperature incubation. So last but not least, we have the mechanical lysis. Here the major advantages is it can be very powerful and fast as well. It's a very good method, for example, for metagenomic studies where you need to make sure to lyse with a minimal bias a lot of different microbes with the same efficiency. Some of the disadvantages here, it's quite complex because I always have an interaction of my sample, my tube, the type of bead I'm using, and the machine I'm using. So I have a lot of factors that I need to adjust. And if I do it in the wrong way, this can also lead to fragmentation of the RNA. So especially if we think of we do a too powerful or too long beading process, it can get too hot, and there's always the chance that I will actually fragment my RNA in that process. Yeah, so how basically does actually the RNA stay alive once it's released from the cell?
Well, there are more issues. After the explosion, the RNA is out in the free and not protected. Whoa, this looks scary. So-called RNases are omnipresent, hunting our little friends and harming them. Wait. Our story has taken a nasty turn. That's certainly not what scientists looking after RNA want. Let's go back to see if there is an alternative storyline. How can we rescue our little friends and make sure the RNA stays intact while we lyse the cell? Oh yeah, that looks better. Adding RNA protecting agents to the story, our RNA will not be harmed. The nasty RNases are denatured, preserving our friends.
Yeah, so as we nicely see, this is basically the next challenge that comes along for our RNA molecules as the cells are lysed and they are released outside of the cells. So RNases depicted by these monster scissors are pretty omnipresent. They're on our skin, everywhere, on your bench, on the pipette tips, and so on. And these are really nasty enzymes. So actually, this comparison with monster scissors comes quite close, as they're really effective and quite difficult to stop. And therefore, we want to really highlight that one of the most important things when you do an RNA isolation is while you are sampling your sample and you store your sample, to make sure that your RNA is protected against these RNases. And there are several factors how we can positively influence RNA integrity. So below, basically, you see different tape station profiles, which show you differentially degraded RNA from nearly totally degraded RNA on the left side to quite nice and intact RNA, and also indicating this Win value, so the RNA integrity number, which gives us an indication how intact our RNA is. And one thing to make sure is really to correctly store your samples at low temperature. So if you're not directly using the sample, please make sure that either you directly freeze them in liquid nitrogen, or you use some protective buffers like RNA later, for example, to store them maybe also at room temperatures if you don't have a -80 or liquid nitrogen available. So this is quite important and will ensure that you can get very nice and good RNA integrities. While on the other hand, you clearly want to make sure that you don't have any elevated temperatures, don't leave your samples on the bench, make sure the time from sampling to put it in the buffer will be fast. Freeze-thaw cycles is also something that will negatively impact the RNA integrity. So once you freeze them, you shouldn't thaw and refreeze them all the time. And then, as I just said before, also the homogenization time and power can be quite critical. So make sure not to use too long homogenization and too harsh homogenization to really make sure you have a gentle lysis and that your RNA is protected. So to give you just some practical examples of how that would look like. So this is some in-house data we did with our protective buffer, which is called Purify Later. Compared that to RNA Later, which probably many of you know, and to a reference sample that stayed at room temperature. And you can see if we stabilize the samples at room temperature and we have to look at the Win values after different time points, so 2 weeks and 1 month, we see that the reference sample at room temperature quickly degrades, while even at the room temperature, using these protective buffers, we can make sure that even after months, we still have quite decent RNA integrity of Win values around 8. And this is even getting much better if we have low temperature. So this really nicely shows that even, just lowering the temperature can have a major impact. And we really would encourage you, before you start your experiment, to think about how you would like to sample and store your samples. Okay, so if we made sure that everything is nicely sampled, nicely stored, so how would we actually go on to extract and purify our RNA?
Well, suddenly our RNA friends are pulled away from all other parts of the cell. They are bound tightly and waiting for what's coming next. The scientists want to get rid of all cell debris, which means washing. In typical RNA extraction stories, our RNA molecules experience several washing steps, during which all other cell components are being removed, leaving our RNA friends isolated as scientists need them for further analysis.
Okay, I really like that animation. So this brings us basically to the second quest along the way of following our RNA from the cell to a tube, which would be the nucleic acid purification. Which is basically the task of separating the RNA and clean it from all of the other molecules that we have in the cell. And talking about this, we have another question to the audience. So we are fairly curious. So what you usually use in the lab, or what are the methods that you prefer to use to do your RNA isolation? So is that rather a silica based bind wash elute workflow? Are you using magnetic beads to do your RNA isolation? Maybe some of you already tried our ecolution technology, or are you rather using really a homebrew method where you mix all the buffers yourself?
I'm curious, Tom, why don't you tell us a little bit about your experience? What did you used to do in terms of extraction in your active time in the lab?
When I did most of my extractions, I was doing my PhD, because I did a lot of RNA-seq. And as I'm a plant scientist, I work with a lot of very challenging samples. And so back then, the best option I had was basically using Trizol for all these plant types, because it was quite universal and a quite powerful lysis, which was nice, but it was not very nice to work with that. So going under the fume hood, it was super nasty. I remember pretty well one time, one of my nitrogen cooled reaction tube just popped up and there was Trizol everywhere. And I had to go under the shower and so on. It was a pretty bad experience. So I
Yep,
would
can
have
imagine.
wished to have some great other kits available, definitely. Okay,
It's good that there are
so
alternatives. Okay, yeah, let's.
we have some results here, so we can push them to the audience. Okay, yeah. So seems most of you really work with silica-based extraction kits. I also believe this is probably the most common method, especially if we do, let's say, in a lower throughput single isolations. Yeah, but there are several methods out there, and we would just like to go on to highlight some of the key advantages and disadvantages and discuss how we can really extract our RNA. So there are various different principles to separate RNA, and we can actually categorize them in three major categories. The first one is really a selective binding of our nucleic acids. So in our story example, this was depicted by the magnet, but the silica would also fall in this category. So here we really make sure that we bind the RNA to a solid matrix, and then we try to wash away all the other components that we don't want to have. There are methods using filtering or size exclusion technologies. And there are also classical precipitation methods, like when you use phenol and a chloroform method, where you would precipitate your RNA and you have a pellet, and then you discard the supernatant and go on from there. So let's take a closer look and stay with a method that we also faced in our storyline, which would be the magnetic bead-based method, because this relies on a classical bind-wash-elute workflow, and we just saw from the poll that this is what most of you are basically using. And the basic principle of this is that we need to use specific binding buffers, and these are usually using either high chaotropic salt concentrations, or they use organic solvents like ethanol or isopropanol, and they really make sure that they rip apart the hydrogen shell of your RNA and make the RNA negatively charged by the phosphate backbone, so then it can bind to the magnetic beads. And then we, of course, need to get rid of these salts, of these organic solvents. So usually we end up having several wash steps to make sure to get rid of all contaminants. And for mag beads, we also have a drying step. And then we can elute our clean RNA. There are several advantages of that method. For example, with mag bead especially, it's pretty flexible, right? So I can adapt the number or the amount of mag beads to the size of my sample. And this, for example, is a method that is fairly easy to automate with many automation solutions available. But it comes with some major disadvantages, which is mainly the purity issue. So we have a very high chance of carrying over ethanol if the handling is not really perfect and accurate. And it's quite tedious, especially if you do manually, doing all the wash steps. Therefore, yeah, it's really difficult to make sure that we don't have any PCR inhibitions in the end here. And this is basically some of the disadvantages that traditional nucleic acid purification methods have in common. So while they are very solid methods being out there for the last decades and very well established to deliver good RNA, this really comes at the cost of doing that very tedious, time-consuming, doing many steps, going from centrifuge, force, and back, many times working with toxic substances. And I'm sure that all of you that use these methods will also face some time where you end up with a lot of plastics and a lot of buffers. So also these are not the most sustainable methods. And so basically what we do at BioEcho, and as Christiane introduced in the beginning, we try to really question the traditional methods on the market and find out if there are some different ways to use and some different technologies to use to do or to enable you easier RNA isolation.
Yes, luckily, again, there is an alternative story that we can tell. Imagine a way to not bind the RNA molecules, but rather hold back all unwanted components of the cell, leaving our RNA friends isolated as we scientists want them to be for our research.
was still muted. So as you can see in that nice example with the slide, yeah, we developed a very novel technology, and this graphic here is quite simplistically depicting that, and I would like to explain you the key differences that we have with our ecolution technology that we developed at BioEcho compared to other traditional methods. So obviously in the beginning, we need to lyse our cells, but here we have a first small difference. So because you remember I told you that for binding your RNA, you need to get specific binding conditions using salt buffers or organic solvents. This is something we don't need as we don't bind. So basically we can really use a lysis that is very targeted and very gentle to the sample type we are using. And then what happens is that we apply our lysate to our purification matrix, which can come in different formats, single spins, tubes, and plates. And what happens is that the nucleic acid, so the RNA, will basically flow through that matrix without interaction, so no binding. And we have a clean RNA in our eluate, while all the other cellular debris, like proteins and polysaccharides and so on and so forth, will be held back by this matrix. And as you can imagine, this brings along several advantages. So you can see again a comparison with a classical bind wash elute method. So now depicted here, classical silica method. And what we can achieve by using our ecolution technology is that we basically combine all these bind, wash, and elution steps in a single step purification, which is basically a 1-minute centrifugation step, which makes our technology extremely fast. So depending on the kit and protocol you use, it's about 3 to 10 times faster. And it also has several other advantages, such as making your extraction much more sustainable, which basically brings us to our next poll question that we would like to ask the audience. And we are quite curious if you're already making some efforts to reduce the plastic waste and the associated consumables when you're doing your RNA extraction, and what would be the main driver that motivates you to do so. Are you already doing so, and sustainability is a quite important topic to you? Or are you rather looking for reducing plastic components to save budget? Or is this maybe something that's not even relevant for you?
Yeah, I'm really curious to see the results of this one, I must say, because, well, sustainability is on everyone's mouth, but let's see how far institutions are or laboratories are in implementing it. And also, it's interesting to see if people are also aware that there is an economic factor to it. So that's maybe
yeah, I'm quite
more
sure.
time.
At least for me, it was pretty obvious being in the lab. You know, ending up every day with at least one, maybe more, completely full of these smaller bench trash, trash cans. So I think it becomes quite obviously.
True.
Yeah, and we see here, we can push that to the audience. Also for many of you, it's already quite important topic, but we can also see that many of you still think that there could be some room for improvement here. And for those of you, maybe if you haven't heard about ecolution technology, that would be one way for you to achieve a bit more sustainability in your lab. As we just discussed, our technology comes along with multiple benefits against traditional workflows, which are nicely depicted here by these illustrations. So first of all, we try to stop any toxic relationships. So we try to work with buffers. You don't need to go under the hood. You don't have any stinky beta-mercaptoethanol, which comes in many RNA kits. There's no something similar to phenol or triazole, so you can safely just work on your bench, and you can just feel safe working with our kits. We skip all the washing steps, so also we don't have that much centrifugation, which is also a shearing force on the RNA. So it's in general a more gentle isolation process as we don't bind. And of course, we save a lot of plastics, being illustrated here, saving a lot of tips. But of course, tips are not the only thing we save. And to make that a bit more visual, we want to give you or show you a comparison here. So we compared on the left side one of our Ecolution kits for 50 reactions against the classical silica-based kit. And you can directly see on that picture, you save a lot of pipetting tips, collection tubes, but also you save a lot of buffers, right? So your liquid waste also drastically goes down. But yeah, seeing basically all of these different advantages, I think many of you will probably be a bit skeptical. I would be as a scientist, so that's what we're trained for. And one of the major questions, of course, that also was the most important for me in my lab life is how does the RNA look after using the products, and how is the performance? And of course, this is the most important factor. So be assured when we develop our kits, we make sure that they really are on the highest level of performance. Of course, we compare them to other market-leading kits. And this is just to give you an example of this. This is our RNA kit for cell cultures. And this is actually not data that we created, but data that was created or results we got from one of our beta testers. And yeah, so we compared the bioecho method to a silica-based kit. And you can see that you get very nice RNA yields, a very pure RNA. And also if you look at the RNA integrity number, so we have our Ecolution method here, and compared to 2 different other suppliers, you can see that we get very intact and non-fragmented RNA, which is really ready for all your downstream applications, such as qPCR, sequencing, NGS, and so on. Maybe one more important thing here, you see the small asterisks. So our kit is also capable of isolating smaller RNA fragments. This is something where many silica-based kits have a cutoff, usually around 200 nucleotides. So you see that with the one competitor here, which these kits have a bit more issues of isolating also smaller RNA molecules. Okay, now we saw, okay, we can achieve a great performance, we can save plastics, we can save time, but to the end of the presentation, we still want to give you a bit better picture and also let you know how does that eventually translate to the costs that you have, because we all know budgets are also quite important for everyone. So to really give you some numbers, and we know we like numbers and scientists like numbers, so we really try to do a case study here where we ask the question, so how does it look if we would isolate 1,000 RNA samples and compare this isolation if we use a plate-based bioecho kit versus a plate-based silica kit, and basically calculated the amount of tips that are used, the amount of liquid waste that is used. And in this example, we looked for automated purification. So also for those of you who are interested in that, of course, it's possible to automate our kits, and we are happy to discuss that with you. And you see, again, saving a lot of plastic consumables, but this also translates to saving costs, right? So there are a lot of associated costs for consumables, for disposal costs, and also, of course, the time that you just spend in the lab is a cost factor in the end for the lab. So in total, also, yeah, calculating in the price for the kit. In this example, we would be able to save around 30% using the bioecho method. So it really has a lot of yeah, additional benefits and advantages of using our Ecolution method. Okay, so with this, we are already at the end of our presentation. So yeah, happy RNA, happy end. We really hope you like this kind of more interactive type of storytelling and webinar. So please let us feedback there if you like it. I would just want to end the webinar with some important, let's say, conclusions or take-home messages. So to really get these happy RNA molecules in your tube, one of the most important things is really to have the right storage and sampling. So this is the main key. So because what we say is trash in, trash out. Once your RNA is degraded, no kit can bring it back, right? So this is the most important factor. Then depending on the sample type you use, make sure to tailor the lysis to the sample type. Think about what could be the best methods to homogenize and lyse your samples. So usually if you end up with a low RNA yield or low concentration, the reasons for that lay in the homogenization and the lysis part. And whenever you have questions to that, feel free to contact us or ask now in the Q&A. Last but not least, we want you to have fun and save plastics and so on, isolating your RNA. So whenever you're interested to test one of our kits or get more information, yeah, just please feel free to contact us and we are happy to consult you. Good. So with that, we come to our Q&A session. So I think we stayed fairly well in time and we definitely have good time now to really answer some of the questions you may had in connection to RNA isolation.
Yes, so we did get a couple of questions. However, still feel free to post any into the Q&A box that you might have. Just in case we're running out of time, we will definitely get back to you within the next days to answer all your questions. But yeah, let's get started with the first one. Yeah, it's towards lysis. So how does homogenization by grinding affect RNA extraction?
Yeah, I mean, I already briefly touched that issue, but this can be quite important, and we frequently see that when people are getting issues with the RNA isolation, that could be connected to, yeah, just choosing a too harsh homogenization. So one factor is temperature, right? I mean, you can imagine if you have a small tube and you have beads, moving fast in the tube, you get very hot temperatures, right? So if you don't have the chance to cool down your samples while doing this bead beating process, I would really recommend to do shorter cycles and between the cycles cool down your samples again. And the other one is the pure shearing force. So if you do it for too long or at too high speeds, you can clearly see if you look at the tape station and you do bead beating, you get smaller RNA fragments or DNA fragments compared to, let's say, enzymatic or chemical lysis.
Right, thank you. Next question would be specific for cell culture RNA extraction. It is, I typically have low numbers of cells. Do you have any tips how to optimize RNA yield?
Yeah. So of course, it's quite important to, or there are many possibilities to adapt the kit to the number of cells that you have. On the market, there are specific solutions for using with low numbers of cells. Using our products, it's definitely possible to adjust the lysis volumes. So we have customers working with very low numbers of cells. So let's say just 1,000 cells or less. And there it's really needed that you really lower down for our method the amount of lysis buffer used so that you make sure you get a decent concentration. Really, if you have a certain scenario, if you know your number of cells, feel free to contact us so we can give you the tips and tricks, how to work that out and really adapt the protocol to your needs.
Next question, I believe, was asked when you showed us the tape station image with the RINs or the different RIN numbers. It is, how is the RIN related to the rRNA ratios, so 28S versus 18S?
Yeah. So, although I cannot explain the exact algorithm behind that, basically the win value is taking, or the win number is taking the ratio of these two peaks of the larger and smaller ribosomal subunit, right? So, which is quite interestingly, because it doesn't really say something about how much messenger RNA or how much smaller RNA you get out there. Basically, this is maybe also an important thing. If you look for different RNA species, like microRNA, we definitely encourage to also use specific tape station protocols for that, right? Yeah, but basically, it's important to get a certain ratio of these two bands, because it's just shown that if you have degradation of RNA, these bands are not that sharp, so you don't have a sharp peak, and then these win values go down and give you good indication of, yeah, if your samples, for example, are good enough or make sense to send to sequencing or not. But it also varies a lot for sample type. For example, typically plant samples have lower win values if we compare that to just cell culture.
Yeah, that's true.
For animal cells or something like that. Yeah.
We got a couple of questions on, well, extracting different types of RNA. Let's start with the first one. For us, the advantages of trisol precipitation is that we get all types of RNA, so mRNAs, microRNAs, long non-coding RNAs, etc., from the sample. So they all can be investigated from the same sample. How is your technology performing in this point of view?
Yeah, very, very nice and good question. And this is very much connected to the very first slides of the webinar, when we say it's important to think about, what kind of RNA molecules you're looking for. And so we consider our isolation technology to be a total RNA isolation, including smaller fragments as well. So we can see in the tape station profile that we have a pretty pronounced peaks, especially for smaller RNAs like long non-coding RNA. We have also been able to detect microRNAs in our eluids. Although I would like to say here that it's not a, let's say, dedicated microRNA kit that we specifically developed to enrich for microRNAs or something like that. But we definitely get the complete different fragment sizes and lengths. So I definitely encourage you to give it a test and see if it works for the application you're looking for. Yeah.
Mm-hmm. Yep. Then we had one person asking for a specific cutoff in terms of RNA size, and one other question that is a little bit more detailed. First of all, thank you for the interesting talk. What is the shortest RNA that can be extracted with the Ecolution kit? Is smaller or larger RNA more or less likely to be retained in the matrix? And how much of the RNA is typically retained in the matrix in percent? Maybe we answered the first part of it first. So
more tricky question. Yeah, so the first is easy. I mean, we don't have a clear cutoff, right? So this typical cutoff of 200 nucleotides below, we clearly don't have. So it's not a black and white kind of story, right? As I just said, we get these smaller fragments, and at least I don't have the numbers to specifically tell you how much percentage the different fractions have. I believe the smaller we get, there is a certain effect that we I mean, as I said, we don't have an enrichment for microRNAs, so there is definitely the chance that from a certain size, the recovery gets lower. But we would need to consult back with our R&D, so I don't have exact numbers on that. Yeah.
Yeah, I think we could get back to that later on. What if we need to extract RNA, DNA, and protein from one sample? Can we use this one-step purification process? Does it work for DNA and protein extraction as well?
Yeah, very good question. So far, the kind of products that we have available are usually really focused on either RNA or DNA isolation. However, we had already many different customer projects where we also looked into simultaneously RNA and DNA isolation. And that also worked out with a bit of adaptation of the protocols. So it depends a bit on what kind of sample and what kind of application you're looking for. One thing that does not easily work is protein. Usually what you do if you have a silica, you just get the first flow through, which has a proteins, and then you can kind of clean that up or participate that separately, and then you can go on with the bound RNA. This is something based on the technology we have that we don't, or that we are not able to do because we do not selectively bind, right? So there's not a very specific fraction of proteins that we could elute at a specific time point. So yeah, getting simultaneous proteins out, I would say, would be quite difficult.
Mhm. How much difference does single or twice overnight incubations make to our final results in trisol method? So single or twice overnight incubation. What if we do not add that overnight incubation and process the samples?
So the question is overnight incubation versus just an hour or a shorter incubation, or did I get it right?
I think it is one overnight incubation versus twice, if I get it right.
Ah, twice. Okay. um, that's very sample dependent, I would say.
So the person is working with uterine fluid samples.
Mhm.
Very specific, I guess.
This is quite specific. I haven't worked with that sample before, but for many samples, it's possible to tweak the lysis in a way that you do not have to do overnight incubations. And I think it's, at least I can speak for our kids, also for the more challenging samples. We can usually get very fast lysis times. That has several reasons. One of them being, as I said, we try to design our lysis buffers very specifically for the sample type. So we have some kits working with enzymatic lysis. So in these cases, it helps us that we do not need to have these kind of binding buffers, which have these high salt concentrations, because this obviously will inhibit your enzyme as well, right? So enzyme digestion works quite or much better if you have a buffer that's really the right reaction buffer for your enzyme. So we can clearly cut down incubation time there. And then also we developed a very novel chemical lysis, which we called ultra-fast lysis, that we use in many of our kits. This is really only taking 5 minutes. Yeah, it's definitely worth maybe to contact us, and then we can see if we can use one of these lysis principles for your cells. And otherwise, also maybe something that's interesting, and what some of our customers are doing is, if you kind of fix the trisol because it's the only thing that works for a sample, obviously our technology can also be used to clean up the aqueous phase of your trisol isolation in a very quick and efficient way. So it's really just taking the aqueous phase, putting it on a purification column, centrifuge for a minute, and you're done. So this may also be an option to save time and resources.
All right, let's take one last question, and this is special for you, Tom, as a plant specialist. What if you're using dried plant material? And I guess it's about tips and tricks for lysis.
I mean, I would say it's definitely good idea. So first of all, drying the samples is also another way to preserve your RNA. If we think about freeze-dried samples, for example, and it has one major advantage, and this is that usually if the sample is very dry, it homogenizes much better, right? I mean, if you have a totally dry leaf and you just touch it, it usually falls apart, right? So it's much easier to, by beads, do a really fine powder and have a good homogenization. The only thing to really consider when using dry samples is obviously plants just have a lot of water, right? So you would definitely want to go down with the input amounts you use compared to the input amounts in the kits, which are usually given in fresh material. So this is definitely a recommendation then to use a bit less material to not just overload the isolation method.
All right. Thank you, Tom. And yeah, thanks again for everyone for submitting such great questions and high number of questions. I think it was a great discussion and lots of fun for me, at least. So I think everyone hopefully enjoyed it. Tom, maybe some final words before we hand over to Christy again.
Yeah, thanks a lot, all of you, for participating. I really liked it as well. I think it was also for us the first time doing such an interactive webinar with such a storyline. I really hope you liked it, and it was a bit fun of entertaining for you. Please feel free to contact us if any questions are still not properly answered. Be sure we will get back to you within the next days. Given the number of questions, it may take a few days, but be sure everything will be answered. And yeah, thanks a lot for your attention and participation.
Well, I'm going to come back on here. Thomas, Christiana, thank you for this delightful webinar. It indeed is so fun to go through this purification journey with you. And as always, it's a privilege to host webinars with the team at BioEcho. Now, before we go, I do want to thank our audience for joining us today and for their interesting questions. As Christiana mentioned, we had lots of questions come in, but we don't have time to answer them all today. So any of those questions we didn't answer today, and additionally, those that are submitted during the on-demand period, they will be addressed by our speakers via the email address you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's fun live event. Now we invite you now to partake in our survey. You can now click on that survey icon on the black vertical bar on the left of your screen, but it will also pop up for you when we close. We would be grateful if you spend a couple minutes answering it. It will help us learn if we have covered your expectations today, and it will help us improve your experience in our upcoming webinars. As always, Christiana, Thomas, and the entire BioEcho team, it's been fun and it's always a pleasure. Until next time, everyone, take care. Have a great day.
Bye-bye. Have a nice
Thank
day.
you. Bye.
Thanks.
Bye.